And I would now like to pass you over to Elizabeth Robertson, Director of Research at Diabetes UK, who is chairing the Robert Turner Research Impact Award. Thank you.
Welcome, and many thanks to all of you who have joined us. This evening, we will be awarding the third Robert Turner Award for Research Impact. This award recognises the significant contributions of UK diabetes researchers in driving forward our understanding of diabetes for the benefit of people living with diabetes and those at risk. As always, I would like to thank Professors Rhys Williams and Steve Bain for proposing this award in memory of Robert Turner, to whom we are all indebted for the groundbreaking UK prospective diabetes study. This year, we are recognising the achievements of an esteemed colleague who is sadly no longer with us. The award will be made posthumously to Professor David Dunger. Professor Dunger was Professor Emeritus of Paediatrics at the University of Cambridge. He received training in paediatric endocrinology at the Hospital for Sick Children, Great Ormond Street. Between 1986 and 2000, he was consultant paediatric endocrinologist and later Professor of Paediatric Endocrinology at the John Radcliffe Hospital, University of Oxford. In 2000, he moved to Addenbrooke's Hospital, University of Cambridge, to take up a chair in paediatrics. David was internationally renowned for his research into the pathophysiology of type 1 diabetes and its complications in children and young people. His studies of genetic and environmental interactions during childhood growth have helped in determining risk for type 2 diabetes. He published over 600 peer-reviewed papers and led major international research consortia. Professor Dunger was a Fellow of the Academy of Medical Sciences and recipient of numerous awards. I will personally miss his gentle, expert leadership of our Children's Diabetes Research Steering Group and the Type 1 Diabetes Immunotherapy Consortium. David sadly passed away on the 20th of July last year. This evening is a celebration of his life, achievements, and the legacy he leaves behind from the impact of his research on the lives of future generations. We are honoured to be joined by David's widow, Dr. Dame Jane Roberts, who will later receive the posthumous award from the chair of our Board of Trustees, Dr. Carol Homden. Firstly, we are going to hear from Professor Ken Ong and close colleagues of David's about his research and clinical impact, followed by personal reflections from just a few of David's colleagues, friends, and family.
Thank you, Elizabeth. When I was asked to assemble a lineup of speakers to illustrate David's research and impact, there were literally dozens to choose from. So I've chosen a handful of colleagues in Cambridge who worked closely with, and in most cases trained under David for many years. I myself have had the great fortune to work with David for 25 years, starting as a clinical research fellow in Oxford. And in 2000, we moved from Oxford to Cambridge, where I soon took up a clinical lecturer position under David, and until my consultant level appointment in 2005. So my entire research training and clinical specialty training was under David's supervision. Most of my research in Oxford was in collaboration with the ALSPAC study, where we described the unusually large variability in growth in infancy. And we showed that rapid infancy weight gain was not simply transient puppy fat, as most people thought at the time, but has significant consequences on later obesity, insulin resistance, and puberty timing. And these findings helped to shift the Barker hypothesis, the fetal origins of adult disease, to the broader DOHAD hypothesis, recognizing the role of postnatal milestones on later life disease and mortality. And this work has had significant impacts on the clinical recognition of overweight in infancy and on the spawning of multiple interventions to prevent infant overfeeding and overweight, including our own baby milk study and more than 20 other randomized controlled trials around the world. As well as the impacts, David had a particular fascination for the underlying biology of fetal and infant growth, particularly the genetic and epigenetic mechanisms and how these link to later diabetes risk. And so when we moved to Cambridge, a key work was to establish the Cambridge Baby Growth Studies, which included far more detailed infant phenotyping and sampling than had been available in ALSPAC. And I acknowledge here the key role here of the late Carlo Acciarini and of our study nurses who've worked on CBGS for the last 20 years. Imprinted genes, where genes that are active either from the mother or the father, bring together the intersection of genetics and epigenetics. And David was fascinated by the concept that fathers can influence mother's metabolism through his genes acting in the fetus. And these highly robust associations with maternal glucose were achieved by leveraging together Cambridge cohorts with an international HAPO cohort with Bill Lowe and Northwestern in Chicago. And as a trainee, I was amazed that David seemed to be best friends with so many investigators around the world. In time, I realized that collaborators warmed to David's enthusiasm, his hard work, and generous nature. And I'll just end by pointing out David's key insights on the adolescent transition, which have directly influenced current practice. As well as being fascinated by biology, David was driven by a genuine concern for his patients. And I'm so grateful that that concern extended also to all of us who are fortunate to work with or be trained by him. And with that, let me hand over to Roman Hovorka, Professor of Metabolic Technology in the Department of Paediatrics and the Institute of Metabolic Science.
Very much, Ken, for passing the button to me. I'm thrilled to be here to celebrate David's achievements and also his legacy. I'd like to take slightly different tack, and rather than focusing on scientific achievements, is to highlight David's ability to bring new or look into new areas and also embrace these areas. Little bit of history about myself. I arrived to Cambridge in 2004. It is when Ken and David's team have been primarily based at Cambridge, and I joined the group at the Department of Paediatrics. At the time, I was non-clinical. I'm still non-clinical, and I was helping David to work in the intensive care, where he also had an interest in managing glucose control. But my background was really in the decision support systems on helping people to give the right amount of insulin. But the opportunity arose in 2006 to work on an artificial pancreas, at the time quite unknown solutions or treatment options for people with type 1 diabetes. We had an interesting discussion with David at the time. It was quite new for him, and I was relatively new in this space as well. What arose from this discussion is that David was very willing and important senior colleague to make things happen for us in terms of artificial pancreas development. So what do you want from a senior colleague? At the time, I was pretty junior and coming with new ideas. David was willing to listen, and I just want to highlight the importance of this ability. David has his own research program, and this artificial pancreas was relatively new, but he was willing to consider it and engage in discussion. He was willing to take risks with us as well. As I mentioned, at the time, I was relatively unknown quantity. David had a unique knowledge of managing clinical trials. He had a very good experience how clinical trials could be designed, what control group should be, lots of additional aspects related to make things happen. And this was one of the key aspects why we were able to progress in Cambridge so progressively. David provided support. He had his own research to do, but he was also willing to help us. And importantly, he was willing to trust. We started doing studies in the pediatric population. This is where we started with the closed loop system, and David was willing to put his name and trust into working in this field. So what is the artificial pancreas? You might have seen a very timely press release which happened today, and there was lots of BBC announcement about the NHS pilot. It's a combination which consists of a glucose sensor, algorithm, and also insulin pump, which reproduces the glucose control by healthy pancreas. And as was being introduced today, it is a treatment which transforms management of type 1 diabetes. It is also happening right now. We are expecting that this treatment will be approved there will be a new policy by NHS, by NICE. But I just want to say this goes back 15 years, and some of the work which we've been doing over the last 15 years has resulted in these achievements. So what has been done over the last 15 years? At Cambridge, we were the one who started working in the pediatric population for the artificial pancreas. Again, thanks to David, who was willing to trust and help us to support in this area. We did studies in the older children, adolescents as well. We have ongoing studies in type 1 diabetes, newly diagnosed children, adolescents as well. And we demonstrate the benefits of the reduced glycated hemoglobin, increased time in range, etc. And I just want to say is where we are currently with closed loop systems in the UK and worldwide as well, results from David's visions and ability to join and lead the clinical aspects of the development in these populations, changing the perspective of the technology adoption and in the clinical environment in the NHS and more widely. And with that, I'd like to pass the talk to Ajay, who will be speaking about his contribution or his linkage with David. Ajay.
Hi, thank you, Roman. So I'm one of the consultants in pediatric endocrinology and diabetes in Addenbrooke's Hospital. I worked with David about 12 years ago and started off my career as a research fellow working for him. And I have been working with him for the last 12 years. David was always interested in understanding the mechanisms of human physiology, particularly the metabolism. And we designed a few very complex physiology experiments. My focus was on growth hormone, IGF-1 axis, and glucose metabolism. I wanted to briefly give a brief of growth hormone IGF-1 axis regulates metabolism according to nutrient supply. And during fasting, it promotes lipid oxidation, which helps to adapt to fasting, whereas in fed state, it promotes growth. These rather opposing actions may be linked to the pathophysiology of some of the metabolic disorders. We already know that low birth weight is linked to reduced height, reduced beta cell function, and increased risk for type 2 diabetes, so-called developmental origins of type 2 diabetes. David felt that these abnormalities are a manifestation of a phenotype which is adapted to fasting, and growth hormone IGF-1 axis may mediate some of these changes. We tested this hypothesis by studying adults on a prolonged fast. We found that adults with low birth weight have reduced insulin levels and a more adaptive fasting response characterized by a efficient energy expenditure during fasting. We also found that healthy adults with low IGF-1 have reduced insulin levels, reduced beta cell function, and increased fat oxidation, particularly in the skeletal muscle, all consistent with a phenotype adapted to fasting. We then took some of these ideas forward to optimize growth hormone treatment in small 4 gestational age children. In a clinical trial, we showed that achieving a higher IGF-1 improves the growth, and that was without adverse effects on glucose metabolism. And it was an important point to optimize the growth hormone therapy in these children. We also looked at the predictors of the response to growth hormone and found that the gene scores for insulin resistance were linked to a poor response to growth hormone. We took this idea forward to design an ongoing RCT using metformin to improve the insulin sensitivity in these children. We also looked at the growth hormone IGF-1 axis in adults. Adults with growth hormone deficiency have an increased cardiometabolic risk. And we found that the IGF-1 levels were very low on growth hormone replaced low lean body mass and reduced and increased HbA1c. And these we should optimize the dose of growth hormone treatment in these children, in these adults. We also looked at young adults with type 1 diabetes who have high growth hormone secretion and insulin resistance. We showed that the use of pegvisomant, a growth hormone antagonist, improves the insulin sensitivity, particularly at the hepatic level, and reduces the intramyocellular lipid. Both are favorable responses in a patient with type 1 diabetes. And this kind of suggests that pegvisomant may have a role as an adjuvant therapy in type 1 diabetes. So these are the main studies we were involved in, and in summary, we tried to evaluate the glucose metabolism in relation to growth hormone and IGF-1 axis. And with that, I want to pass this over to my colleague, Loredana.
So good afternoon, everyone. I'm Loredana Marcovecchio. I'm an academic consultant in pediatric endocrinology and diabetes in Cambridge. And I had the privilege of having David as my academic supervisor and also working with him on several research projects since 2005 when I joined his research group. David has been and will always be for me, I believe for many of other fellows who trained with him, a role model for his strong commitment to research, his perseverance, his great contribution to the field of pediatric diabetes. And from a personal point of view, also for being a caring and kind mentor. I closely worked with David on one of his main research areas of interest, which is the early detection and prevention of vascular complications in adolescents with type 1 diabetes. David's great contribution in this field was based on the establishment of several cohort studies of young people with type 1 diabetes, namely the Oxford Regional Prospective Study, which was the first cohort that David set up back in 1986, followed by other cohorts such as the Nephropathy Family Study, which was part of a larger study. The UK grid that David set up with Professor John Todd with the aim of collecting a large number of DNA samples, which then led to remarkable contributions in terms of the understanding of the genetic background of type 1 diabetes. And during more recent years, David set up another important cohort studies, which is the ADIT study, with collaborators across the UK as well as in Canada and Australia. And overall, this cohort have provided important insights into the natural history of vascular complications in young people with type 1 diabetes. And in particular, one key finding has been that adolescence is a critical time for the development of early manifestation of subclinical manifestation of complications, such as, for example, increases in albumin excretion. And this is due to several risk factors, which I have tried to summarize in this slide. And overall, these findings have been important because they've led to the design of the first clinical trial with ACE inhibitors and statins in this population, this young population with type 1 diabetes, in order to try to prevent complications. And this trial was designed and led by David. But even more importantly, all these study findings have had an important impact into clinical practice because have informed, they are still continue to inform national, international guidelines on the management of vascular complication in young people with type 1 diabetes. Another key finding which emerged from David's studies was the identification of a potential new important early marker of complications, represented by subclinical increases in urinary albumin excretion. And back in 2007, David suggested a potential new stratification approach for adolescents with type 1 diabetes based on tertiles, the so-called albumin-creatinine ratio, a way of measuring urinary albumin excretion, with the hypothesis that those adolescents having an ACR in the upper part of the normal range were at higher risk of developing complications. And this was proven to be true. In fact, later studies mainly showed that those adolescents with an increased ACR, in these figures shown by the red lines, were at high risk not only of progressing in terms of renal disease, but also retinopathy, and also some early vascular manifestations, some early vascular changes. And overall, these findings led to the proposal of a new strategy for the screening and management of adolescents with type 1 diabetes, based on the use of ACR as a valuable tool to identify adolescents at risk and implement more targeted intervention based on the levels of risk. And this was some working in progress for David. Hopefully, this will be something which in the future could be implemented into clinical practice. Thank you, and I now pass over to my colleague, Dr. Emile Hendriks.
Emile Hendriks, assistant professor and consultant here in Cambridge, and we've heard so many areas of David's impact already, and I've got three more: C-peptide, Inodia, and immunotherapy. I joined the department in 2016, relatively recently, also as a trainee, and I wanted to start with David's impact as a clinician, and many have mentioned it before me, and his wealth of knowledge and experience in the field, and his keen and eagerness to share that with trainees. I feel very lucky to have trained under David. Also really wanted to highlight his loved by his patients, and show this image on the right here drawn by a patient to represent Prof. Dunger back in 2011. If I then his research impacts start with C-peptide. So David has always had an interest in C-peptide, and recently developed a new technique to look into C-peptide and dry blood spots specifically, which is an easy finger prick test that can be done at home, and it's shown here on the right, kind of what that would look like on a Guthrie card. And this paper that was published a couple of years ago showed that there's good agreement with plasma C-peptide, and that actually there's striking variation in C-peptide between different patients. So some at the bottom have no C-peptide in the first year of diagnosis, whereas others on the right have C-peptide but it clearly declines, or have C-peptide that just stays stable. And David recognized that this could have really important role in maybe monitoring at-risk family members in the future, and also in intervention studies for newly diagnosed type 1 diabetes patients. Next was a new consortium that David was the founding father of, and this was a translational approach and was called an innovative approach towards understanding and arresting type 1 diabetes in NODIA. Anyone that has had a lab meeting with David will recognize a scheme like on the right, where he would use boxes, arrows, and numbering to gather the discussions and his thinking process in a clear overview. And this overview went on to create the Enodia Consortium that consists of 31 academic partners and 6 companies and 2 patient organizations led and founded by David Dunger and Chantal Mathieu in 2014 and kick off in 2015. And the common goal was to fight type 1 diabetes by bringing together leading clinical and basic science researchers, and the first step in this big consortium was a natural history study of newly diagnosed type 1 patients and unaffected family members. I just wanted to highlight some of the achievements of this consortium. So over 700 newly diagnosed patients included in the natural history study, over 5,000 first-degree relatives screened, of whom 342 are in follow-up because of autoantibody positivity, and an ongoing collaboration between the different investigators, and lots of publications over the years, and this is ongoing. And but has then moved on to really final important part of my talk, which is to look towards further treatment of type 1 diabetes and to immunotherapy and to develop clinical trials. And so Enodia is performing mechanistic-based clinical studies, and they're using a master protocol, which was also one of the brainchilds of David, which is established in the Enodia Consortium. And the dream here is to ultimately halt further disease progression. And I wanted to end with just highlighting that over 100 patients have been randomized in these studies and are currently actively receiving a treatment as part of these immunotherapy trials. A great legacy. Thank you.
My little brother David, and he was once, for a while, little to my big, was like most little brothers, very sweet and very annoying in equal measure, but he never failed to impress. Always an intense, focused child, he developed serious interests early on, asking for and receiving a Revell plastic kit of the human anatomy one Christmas, whilst I, 4 years his senior, was still hoping for a model train. David had already decided on his future. And so it was. He worked hard through school, but struggled to get into medical school in the days when who you know often mattered as much as what you know. Eventually finding a place at Charing Cross Hospital Medical School in its historic location opposite the London railway terminus. During this period we were housemates, along with a friend of mine, and Bruce, aka Emmanuel, a close friend of David's, who dossed down for free under our stairs. David and Bruce became fringe hippies, and Bob Dylan, with all his worldly wisdom, was the order of the day. Maybe this enhanced David's often laid-back persona, but nobody should be deceived. David was always passionate beneath that cool and calm exterior, in his work, his beliefs, and his attitudes to social justice. David's notable irreverence for many in authority, for outmoded procedures and constricted thinking in general, was very much a family thing shared by us all. We can't help it, but On everybody's behalf, we will make no apologies. With a career in the airline and travel industries, global travel has been my life. But David must have come close to matching my travels. Like me, David loved to travel. He worked abroad and travelled extensively throughout his amazingly productive career. Certainly he managed to get to several countries and many places I may never see. Do I have a special memory of my brother? Maybe it would be our fights. Childhood territorial struggles later became adult duels over politics. We reviewed from opposite sides what in reality was generally a shared centralist pragmatic view of the world, but differed massively on the remedies. Always well lubricated with whatever medicinal fluids were available, these battles could be memorable, only to be forgotten in the morning. Until the next time.
Good day. My name is Francesco Chiarelli, and I'm Professor of Pediatrics and Pediatric Endocrinology at the University of Chieti in Italy. I would like to thank Diabetes UK for giving me the opportunity and honor to record this short video in memory of Professor David Dunger for his 2022 Diabetes UK Robert Turner Award. I first met David in November 1994 at the International Diabetes Federation meeting in Kobe, Japan. After that meeting, David and I have developed a close friendship and scientific collaboration, which continued for almost 30 years. Several of my young colleagues have been research and clinical fellows in Oxford and Cambridge. In particular, I want to mention Angelica Mon, who joined David's group in Oxford for 3 years in 1995, and Loredana Marcovecchio, who has been in Cambridge since 2005, and she's still there, working in pediatric endocrinology and diabetes, particularly in the field of diabetes-related vascular complications. David visited Chieti many times, since he loved traveling to Italy, like William Shakespeare and Lord Byron. I made jokes with him, and enjoying Italian landscapes, art and architecture, as well as food and wines. I enjoyed working with him and his co-workers, particularly Julie Edge, Angie Watts, Carlo Acerini and Ken Ong, and we have published several papers together. It has been a privilege for me and my close collaborators to know Professor David Dunger, an excellent scientist and a close friend. David has given major contributions to knowledge in pediatric endocrinology and diabetes, and I'm sure that his papers will remain milestones in this field, as his friendship will remain with me for the rest of my life. Thank you, David. Many thanks to Diabetes UK and to you all. Thank you very much.
Well, how to tackle David's career and how it influenced me in a minute is a real challenge. But I think one of the main points that David always got across to me over the years, and what he demonstrated extremely well, was that he could take a clinical problem, and this applied to any clinical problem we might face. He'd look out for the scientific evidence that might solve the problem. And if it wasn't there, what he would do is go out and create it, do the studies that were needed. But he didn't stop there, which is often what happens in many academic circles. He then took that information and then put it into practice. And that's evidenced by lots of work he did creating guidelines, which there are numerous ones over his career, and also the free advice that he was generously offered to all his colleagues. David, I think, was quite a humble person, and he was humble enough to realize that he couldn't do all of this as an individual, and that he needed to collaborate. And whether that was in molecular biology, statistics, epidemiology, or physiology, he didn't mind. He was gracious enough to understand and realize that there are often greater minds than his, if that's possible. Above all, though, I think he wanted, through science and the practice of medicine, in particular in pediatrics, to bring the maximum benefit to the most people that science and pediatrics could touch. And those were his principles of life right through. And he demonstrated those, we talked endlessly about them on many on-call evenings at Queen Elizabeth Hospital in Hackney Road. Those were his deep principles. And he achieved those principles admirably. And rather like Robert Turner, David challenged and asks us also to challenge current thinking, current practice. And we owe to both of them to take that legacy forward. And that a lot should be our living memorial to David's life.
Dear friends, it's a pleasure to join you today to talk about my good friend, David Dunger. I got to know David in 2014 when we came together with the idea to start in Europe a big network where clinicians, but also basic researchers would come together to build a network to do research on biomarker discovery in type 1 diabetes in Europe, and also to start a clinical trial network to come to novel clinical trial design to try and arrest type 1 diabetes. David was a good colleague and became a very good friend. And what I learned from him is that we need to pay attention to all people living with type 1 diabetes, and in particular, the small ones, the kiddies, as he used to call them. David was a warm personality, but an extremely stubborn person. And I learned so much from him. I have him on my phone, and sometimes I still listen to his voicemail. He had such a calming effect on me. I am a very nervous person, and David really knew how to put everything in perspective. He will be missed. He is missed from day to day. And I hope to carry on his legacy on one day preventing and curing type 1 diabetes, and especially prevent the disease in the kiddies. You are missed, David.
This is Dennis Danneman, and I've known David Dunger for the last 30 years. It is entirely appropriate for him to receive the Robert Turner Prize from the Diabetes UK for his contributions to pediatric diabetes research over the last period of time. It's just a pity that he couldn't be here in person to receive this award. To put it simply, David is the most prolific researcher and publisher in the field of pediatric diabetes in the last 25 to 30 years. PubMed lists over 650 papers in his name, and I'm very pleased to say and proud to say that I'm a co-author on 25 of these. I've worked closely with David in the DKA Consensus Conference over the years 2003-2004, and with him in the ADIT trial as it started in its very infancy almost 20 years ago and reached fruition about 3 years ago. David is a tireless worker and could be seen at umpteen meetings off to the side with his sleeves sort of rolled up, finishing a talk that he was about to give, or a paper he was writing, or a grant he was writing. When it came to edit, he always found the bridging funds so that we didn't go down the tubes, and the trial was highly successful. It's been my pleasure to work with him. I miss him dearly. The world is short one superb pediatric diabetologist. Thank you.
Hi, my name is Lynn Ahmed. I was a research assistant in pediatric endocrinology at the John Radcliffe Hospital in Oxford. I first met David in 1986 when he came to take up the post of consultant pediatrician. He appeared very quiet and kind and was easy to get along with. He was not authoritarian, but he obviously had ideas of the way he wanted the unit to be run. And he quietly went about building up a team of doctors and support staff. He was a prodigious worker and led by example, putting in a full day of clinical work, and then evidence of his after-hours work would appear on all our desks the next morning. You felt inspired to do the best you could because he worked so relentlessly and so obviously cared about the children under his care. He constantly was thinking about projects, and writing a paper with him could be both a nightmare as well as a huge learning curve. It often started with a nugget of an idea scribbled by him on a scrappy bit of paper. Then you had to fill in the gaps, and it went back and forth many times with frequent, real cutting and pasting. He let you take ownership of your study, giving gentle, thoughtful guidance throughout, and expecting high standards of patient care and scientific rigor. He was a private, irreverent, and at times shy man. He created clinical research teams in both Oxford and Cambridge, and each individual and their contribution was valued. Before it became a buzzword, he had a knack for enabling team bonding, with many restaurant-themed dinners, wear a yellow tie, a silly hat. We went punting, we had picnics. We went to the dogs literally at the Greyhound racetrack and had parties in our homes with family. We saw each other's children grow up. David encouraged me to do a PhD on the cohort of children with type 1 diabetes, whom I had followed for many years, documenting their diabetes markers as well as their pubertal ones. This wasn't finished until the end of my career, but he was fully supportive of me throughout. He was a friend as well as a boss, although he would shrink at that word. He was a mentor, and I miss him deeply.
Hello everyone. We're sure our dad would have been so proud to have been honoured with this award. For as far back as we can remember, he's worked tirelessly in this field, even to the extent of collecting my pee and keeping it for testing in the fridge when I was little. He so loved his work and his colleagues in the field. He was passionate about finding a cure for type 1 diabetes and helping make lives better for those with this disease. Even after decades in the field, nothing brought him more joy than talking about growth hormones over a glass of wine.
We're incredibly proud of our dad and his dedication to his work. We know it would mean the world to him that you're all here today, so thank you for coming.
Hello, everyone. Well, Diabetes UK introduced the Robert Turner Award for Research Impact 3 years ago in order to recognise the outstanding research impact of Diabetes UK funded researchers. There is absolutely no doubt whatsoever from all of the testimony that you have heard that the work of Professor David Dunger over 30 years is absolutely outstanding and unparalleled in his field. It therefore falls to me as the new chair of Diabetes UK to convey and to all of David's family the huge appreciation of our whole community engaged in the battle against this disease for all that David has achieved, and to recommit the whole of our organisation and our family of friends and researchers to fulfilling and carrying on his work. So Jane, it is with that we send you all our love and appreciation for all that David has made and achieved, and convey this award to you tonight.
Thank you, thank you, thank you. May I show this very lovely award from Diabetes UK? It is really very lovely. So thank you, Carol. Thank you, Diabetes UK, for having chosen to honour David with the Robert Turner Award. It is indeed a very great honour, given the immense stature of Robert Turner. David would be humbled. That's a word that's been used already this afternoon. As David's wife, I'm very pleased to be presented with this award on his behalf, but of course very sad that he is not here himself to receive it. David died, as you heard, July last year, rather too quickly in the end, but it did mean that he could work until very soon before he died, which was something that was very, very important to him. We, that is Daisy and Jack, his children, Peter, his brother, and I, of course, miss him dreadfully, more than we can say. David would have been, albeit very quietly, I have to say, over the moon to have won this prestigious award from Diabetes UK, an organisation that does so much to promote research into diabetes, a pernicious disease, a step change in the delivery of care to individuals and their families, as well as the care, support and advice that you provide. He would have been really over the moon. That being the case, he was not one, as you've heard, to seek the limelight, not to garner awards for their own sake, or claim unmerited authorship. Indeed, our non-medical friends were astonished to learn only after he died, I mean, really, of his eminence professionally. They had absolutely no idea. On the other hand, however, David was very keen that his work, always in conjunction with others, of course, was understood, acknowledged, and crucially translated into clinical practice so that major improvements in the care of young people with diabetes and their families could be made. People referred to him being calm, but you know, beneath that laconic, laid-back appearance, he had something of an aging hippie about him. In fact, he had an awful lot of an aging hippie about him, it has to be said. But he had a ferocious intensity for his work, as you've heard. His ambition was to find a cure for type 1 diabetes, and he was mightily teed off that he didn't live long enough for that to become a reality. It was the quality of David's thinking that first struck me when I first worked with him many hundreds of years ago in a neonatal unit. He was my senior registrar. He was questioning, curious, restless, persistent. And this is a neonatal unit which had quite a kind of cookbook with understandable approach. He always wanted to understand why and think it through, starting from first principles, even revisiting first principles. And that determination to think critically infused David's work throughout his life. I much admired it, and his drive to nurture young academic talent. That was so important to him, that there was rigorous research that was promoted, particularly within paediatrics. Now, although David could be demanding, wholly unreasonable, and exasperating, believe you me, he could, by his own admission, he was never so with patients. With stroppy teenagers struggling with their diabetic control particularly, he's very gentle and understanding. And more than anything, he was gloriously irreverent. I much love that about him. Made me giggle, mischievous with a dry wit. And that he was receiving this award on the 1st of April would have made him smile wryly, and no mean delight. So a heartfelt thank you to Diabetes UK. May you long continue the wonderful work that you do. And importantly also to all of David's colleagues whose work has made this possible. May that continue too. A big, big thank you.
Thank you, Jane. We are humbled and inspired in equal measure, and all of us will do all that we can to take David's work forwards. So it now falls to me to pass back to Elizabeth Robertson to close our session.
Many thanks, Carol and Jane. So many thanks to everyone who's contributed to this award ceremony, and many thanks to all of you for attending. And of course, thank you to Jane and David's family for joining us this evening. So to end, some lovely photos of David.
you.